So with no further ado, I'd like to begin by asking Ben Brown to come join me.
Hi, good to see you.
How are you?
I'm very well.
So Ben is an super overachiever who has many titles, does a lot of different things. He's brilliant and fun to work with. And he has been helping us analyze the work we're doing and explain today to you some of that stuff in breast cancer. Would you care to do that?
Sure, I'd love to.
Okay, let's hear it.
All right, thanks so much, Mark. I'm going to borrow this here.
Yeah.
All right, so I've had the pleasure of working with these guys for the last several months now on the project I'm going to describe today. It's been a tremendous amount of fun, and I'm going to tell you about our experience working with pattern computer to use machines to understand biomedical systems. So my background, I'm a computational biologist. I work on complex systems, whether that's trying to understand how an ecosystem functions, trying to understand the gut microbiome, or trying to model how cells work together or how genotypes work together. Anything really complicated is sort of the domain of the kinds of problems we tackle. And even when you're looking at something as simple as a single organism, a single human, common throughout the human population are at least 6 million genetic variants that occur in 20,000 genes. And even if these variants had individual effects, so that they just operated one at a time regardless of what other genes were doing, that would be a big problem. But of course, genes don't work in isolation. When you look at the possible combinations across genetic backgrounds, across all of these genes, the space of potential interactions among variants is 10 to the 170th. To put that in perspective, there are 10 to the 83 atoms in the visible universe. Okay, so this is a number that can never be explored by brute force. Clever exploration strategies are needed to find the important aspects of genetic background, of genotype, that drive human phenotype. So if you want to do something like precision medicine, like use what we know about the genome to understand health outcomes, you've got a very serious problem on your hands. But of course, it's a problem we'd all really like to solve. We'd love to be able to associate individual genotypes with specific treatment recommendations, or even with unique drugs that promote the most beneficial outcomes possible. Right? So that's a setup for the sort of stuff we're going to talk about today. One of the primary tools we have for doing this, of course, are case control studies. So we've got one individual with, say, breast cancer and one individual without. Or if we're very, very lucky, which rarely happens, we've got before and after, things like this. But we've got a set of data that we can think of as a nominal state, a healthy state. And we've got a set of data where we think, okay, that's a disease state, and then we want to see what the difference is. If you're in a system that is very well controlled, for instance, say you've got a bunch of mice in a cage, you can control the built environment, you can control the food, you can influence their behavior, you can select particular populations. When you do all that, you can test every gene one at a time, or every variant one at a time in the mouse genome, and ask, is this variant associated with some particular state? So this is a study we published last year. This was looking at a particular type of microbe that occurs in the mouse gut microbiome. It's a beneficial microbe, so you want your healthy mices to have lots of it. And what you see here, every tick on that histogram-looking thing, this Manhattan plot, corresponds to a single genetic variant. And then down at these box plots on the bottom, these things show the association between particular variants and how much of this bacteria they have in their gut. And so what you notice, these axes are on log scales. So depending on a single variant in the mouse gut, you can get a 10-fold increase in the amount of this beneficial microbe they have in their gut. So there's a very powerful link between mouse genotype and its gut microbiome, provided that you can control the diet, control the built environment, control everything else. And then that Manhattan plot, the y-axis, is a measure of statistical significance. That black line is a level of what we call genome-wide significance, meaning that it holds up statistically. And you see that some genes, or many variants, pass that threshold of statistical significance. And clearly the effect size is enormous. So, okay, we believe these things are associated with outcome. Now, when we look at humans, it's usually much noisier. because of course we can't control everything. We can't control diet, we can't control built environment, et cetera, et cetera. But people do this all the time. We do these genome-wide association studies, or GWAS, in humans as well. And they've identified now many thousands of genes, in fact, associated with particular outcomes or disorders. There's a problem with these analyses, though, because what they yield fundamentally is not a causal picture of how the disease arises, but a parts list, And unfortunately, that means rarely actionable intelligence. So if you look at the first line in this table, you've got Alzheimer's disease. There are 5 known and approved drug targets for Alzheimer's disease that have, the GWAS studies have revealed 54 genes associated with Alzheimer's. The number of, 5 approved drugs. The number of drug targets, 179, massive number. The overlap between clinically actionable targets and GWAS targets is 0, right? Nothing. And that unfortunately is not unique to Alzheimer's. That's sort of the status quo. Less than 1% of genes identified by these genome-wide studies turn out to be actionable targets in the end, right? They provide some insight into what might be going on, but not so much, and it's not usually actionable intelligence. The problem is that it's just returning a parts list, okay? So this is the difference between knowing all the parts that go into a car and knowing how the car assembles. Knowing all the parts is nice, and you can say, oh, you got a problem in this round-looking thing over there, which is about my level of familiarity with automotives. You got a problem in this round-looking thing. But if you don't know what that round-looking thing does in context and how it works together with all of the other parts that make up a car, you have shockingly little actionable intelligence. And that's the problem. That's the paradigm shift that has to change. We have to learn how to extract structure from the data. As Mark said moments ago, we have to learn how to open the black box. Okay? So that's what I'm going to talk to you about now. So one of the first things these guys did is they asked, can we create better prognostic panels? Okay? There are lots of prognostic panels. It's a huge industry. Lots of folks are making these things. There's the Blueprint, the PAM50. So these are breast cancer diagnostic panels. What you do in these things is you take a sample of tissue and you extract RNA and you analyze it for gene expression. And you ask yourself, is there a unique signature of a particular cancer subtype? You really care about subtype because it's associated with survivability. It's also associated with particular therapeutic directions. Right? So for the major breast cancer types shown here on the 4 columns of this heat map, we've got luminal A, luminal B, HER2, and basal. Right? These are different types of breast cancer. And down the y-axis on this guy, all these different blocks, these are different individual genes. Blue means low expression, red means high expression. And as you can see, when you look at the genes in this panel, you get very different patterns in these genes for the different subtypes. This is very cool, but of course, and some of these are in the Blueprint PAM50, or some of these are in Blueprint, some of these are in PAM50. Many of them are unique. So this has an 89% accuracy. One of the things we're working with them now is trying to validate this, because it looks like if you do repeat sampling on a single tumor, you maybe push accuracy to 99% or above. And that's a subject of interesting research. Another very interesting thing is you can look at.
You said 99%.
Yeah, I said 99%. Yeah, for repeat sampling, yeah, absolutely. So now you can also look at the intersection of subtype and survivability, because while it's true that people with certain subtypes generally have much worse prognosis than others, right? Basal versus luminal, for instance, there are outliers. There are people with basal who are going to have excellent prognoses. Right, and how do you start to differentiate that? So say you've got 2 people, a patient A and a patient B. They both come down with high risk. But now you want to understand more. You want to understand the context. You can start to dive into this in an informative way, right? So this is a probability cube. So this is a summary of data in 3 dimensions. So this is looking at 3 different genes. And this is summarizing prognosis over the entire domain of expression values of each of these genes over a patient population. Blue means good prognosis. You're going to be alive 10 years from now. Dark red means very, very bad prognosis. And you can open up this cube and see where individuals fall within. You can do this. You can take different slices. You can look at different genes. One of the things that they returned to us when they did this analysis on some of the data is lists of these cubes, right, that are prioritized for interest. This one is particularly nice because it has a structure that I call and-like logical structure. You see it proceeds in a sort of diagonal across the space from very low risk in the lower right-hand corner to very high risk in the upper right-hand corner. So if these collections of genes, one transcription factor and 2 genes involved in cell cycle / DNA repair, you see that if all these things are lowly expressed in the lower left-hand corner, everything's sort of groovy. And if all these genes are highly expressed in the upper right-hand corner, prognosis is potentially very, very bad, right? And so that's very nice because now you have a prognostic engine that you can look at, but it's a little bit more than that too, which I'm going to get to in just a second. So just looking at this one interaction closely, so it turns out that this FOXM1, this transcription factor, is known to regulate both of these kinases that are involved in DNA repair and cell mitosis. And once you have those probability cubes, you can just extract a decision rule from those based on the learning machine, and then ask, okay, what are the Kaplan-Meier curves like? What is the differential between survival or dead, right? And so the machine tells you that with these 3 genes, an indicator built on these 3 genes is better than an indicator built on any one of them by quite a margin. And when you look at the joint distribution of all 3 together, you see that you can get a very nice separation between alive and dead. And of course, you can look at the cube and see what the architecture of that looks like, right? And this is a very important thing to know, right? Because it's not just a prognosis, it's potentially actionable intelligence. So this is a representation, another representation of the heat map, another representation of the cube. And you've got our 2 people, A and B. They're both in the red region, so we both say they have a poor prognosis, right? A poor prognosis. But we have more than just a 1 or 2-dimensional view of this. So if you just look at this 2-dimensional slice, okay, they're in a poor prognosis. But if only their level of expression of BUB1, that checkpoint kinase, if only that thing were a little bit lower, you'd be groovy and you wouldn't be so worried. Now I want to animate this for you. Can we roll the animation? So when you dive into the 3rd dimension, you see that the architecture is actually much, much different than it looks when you have just the 2-dimensional picture, which is in turn much different than when you have just the 1-dimensional picture, right? When you look at this 3rd dimension, when you condition on the expression level of check 1, you see that one individual is quite a ways in this gene expression space from anything like a halfway decent prognosis, whereas the other is right on the boundary between poor and favorable prognoses. Now, every single one of these genes has a preclinical drug designed to hit it. What this gives you is a map from gene expression space to what you might want to do to move person A or person 1 or person 2 from the space they're in now to a space with improved prognosis, right? And of course, you don't just have one cube, you have many. You can integrate across these. And that's a really different and really exciting type of intelligence that we frankly haven't had access to before. Okay, so that's my overview of the stuff we've done so far. It's very exciting. I'm very excited to go forward.
Thank you. Thanks. Let's see. So a couple of quick questions, kind of summarizing. It has seemed to us in getting to this point in working on breast cancer, this is all about breast cancer, that we've found a new way to understand cancer, by which I mean how to see it and how to actually manage it in terms of a risk portfolio, which you just described. So that we could say, for the first time ever, maybe we can look at a risk portfolio more intelligently, have a more deep understanding of the risks. And for any given patient, put that patient onto that chart you just showed in 3 dimensions and see how that patient could get out of there alive.
Yeah. That's exactly the hope. That's exactly the hope. And we're visualizing here, these 3-dimensional cubes, which are essentially a 4-dimensional data representation, right? Because we have the X, Y, Z, and then the response, which is color-coded. What we received when we ran this analysis was interactions of order 6 or higher, right? Which we don't necessarily know how to visualize yet. but you can use them in the same way. So we can conduct now optimization. So you can ask, okay, what is the optimal trajectory for this person in terms of risk associated with perturbing gene expression, whatever you want. So even if you can't visualize the data, you can use it to have an engineering level understanding of the system.
Right.
Again, I analogize it to the difference between having a parts list and knowing how the machine works.
Yeah. And last question, I'll let you go. What's it been like for you working with this interesting team?
Oh, it's been a blast. It's a very diverse group. The bioinformaticians are wonderful, and the mathematician in me is very fond of the rest of the crew as well.
Okay. Okay. Thank you. Thank you, Ben.
Thanks, Mike.
Take care.